Hello, this afternoon I'm going to talk about the structure of the DNA and its replication. First of all, the structure of the DNA is the nucleic acids. Those are polymers of nucleotides. Um, the strands of the DNA, the base, and the composition are the DNA is, for example, a double helix. It's anti-parallel. It's what is it called? Sorry, linked by hydrogen bondings, and it gets together with complementary base pairs. In this case, the DNA is from 3 to 5, one strand, and 5 to 3, the other strand. We've seen how the DNA contains the letters, which are the components in there, which is A for adenine, T for thymine, C for cytosine, and then G for guanine. And it gets together this way. A with T, adenine with thymine all the time, and then C with G, cytosine with guanine. And it replicates in something I call the unzipping. Sorry. We can say from here, which is C with G, as I recall in the other example over here. And this little thing, it's a helicase, which is running from this way, copying all the letters, putting them together as complementary base pairs. And at the end, what is gonna happen and is that the original strand is gonna end up copying one strand, which is in this case is a blue one, and it's gonna give us two different DNAs. As we can see, the blue one is gonna be the copied strand, and it's gonna create a new different strand, which I represent in this drawing with the red color. First of all, the separation of the double helix has to separate into two single strands. Then the helix, held together by hydrogen bonds between the complementary base pairs, is separated by helicase, which begins at a point or at an end of the DNA molecule. And it keeps moving one complementary base at a time. This, for example, this is the unzipped section. This is the part that is barely about it replicated, is dividing barely. And the DNA, of course, this replication, its purpose, the conclusion for it, is that it ensures that two identical copies of the DNA are produced from one of the original. Then no DNA molecule is completely new, and the O pair always creates a new strand. It's a semi-conservative process. It's a pre-existing, and it's always the DNA molecule is always safe. Thank you.